Okay, up next we have Matt Foli, and in case you're confused, yes, we did change the order of the speakers. So the third's talk in this room is going second, the second's going third. So you're in the right place? Okay, who's Matthew?
Great. Hi everybody, my name is Matthew Foli. I'm a second-year master's student at SUNY ESF, and today I will be talking to you all about my ongoing research titled Basal Resource Contributions to Young of Year Northern Pike Across Nursery Habitats. So to start off, we can think of basal resources as the carbon that supports our food webs, and in aquatic systems this can be our macrophytes, and also our algae such as benthic algae, phytoplankton, and filamentous algae, our living carbon, but it can also be our once living carbon, including detritus, fine particulate organic matter as well. Basal resources can vary both in time and space, and that variation is the focus of this research. So our study species is northern pike. Northern pike is a cool water apex predator, and they're very much a signal of spring. Shortly after ice out, they will move up the wetland gradient into flooded wet meadow habitat to spawn. They're generalist feeders, and in the Thousand Islands region of the St. Lawrence River, their populations have been observed to be declining. And part of that decline is from loss of spawning habitat. That loss can be explained partially through the creation of the Moses Saunders Dam, with water regulation that can prevent or shorten access to the spawning habitat. And that stable water levels can support Typha X glauca expansion, which is right here. It's a hybrid cattail that is dominating in the Thousand Islands region. That expansion of Typha X glauca can also block or replace spawning habitat, and it also has effects on the environment, in the local environment, such as detrital loading, which can increase hypoxic events. So what does this expansion look like? Well, if we look at this top left photo here, this is Cobb Marsh in the St. Lawrence River 10 years before the dam's creation, and we can see a gradient from the bay into wet meadow habitat, that kind of gray patchy stuff being vegetation. 70 years after the dam's creation, we can see that Typha has kind of taken over that wet meadow habitat. In the photo below, this is Flynn Bay in the St. Lawrence River. Again, Typha has dominated, but we see these green patches here, here, and up there. So there's still wet meadow habitat, but if you look on the photo on the right, Typha makes these thick walls where pike they just can't get through it to access this wet meadow habitat. And so what we have observed, and studies have done in the St. Lawrence, is we're starting to see a protracted spawning period for northern pike. So what that looks like, in the early spring, when water levels are high, water temperatures are cold, the adults will move up this gradient into wet meadow habitat that is available and spawn in that warmer water. And then we'll have their offspring start to develop in that habitat that we'll call a wetland habitat. In late spring, with water management, perhaps reducing water levels to prevent flooding, or it's just a low water year and they just can't access wet meadow habitat. The water is still warming up, so now the bay habitat is suitable for spawning, and we see the adults come in and spawn in these bays. And this creates these two groups of young of year northern pike that we want to focus on. So that leads to the central question of, do basal resource contributions to northern pike vary across these different geomorphic nursery habitats? And we're focusing on coastal bay wetlands and drowned river mouth wetlands. For ease, for the rest of the talk, I'm just going to call it the bay and the wetland. So we have 7 study sites. The yellow ones are the 3 bay sites on Grindstone Island. The red, it must have been missing a wetland, but is our wetland sites along these Thousand Islands. If we take a closer look at our bay sites, we see that all of them have Typha presence, that cattail here, through here, up there, where wet meadow habitat once was. And if we look at our wetland habitat, habitats. We can see typha again is dominating through all of these. All of these were, in the last decade, had restoration projects conducted through these partners, creating these connectivity channels and spawning pools there, all through this site, and here, and through there, in which the pike are now able to access wet meadow habitat, but also use the spawning pools to spawn in. And they have had some success in using these spawning pools. So if we're trying to understand how basal resources get and support the tertiary consumer of northern pike, we got to fill in the gaps and also look at our primary and secondary consumers as well. And so when we went out to the field, we collected all of these through a variety of methods: grab samples, D-net sweeps, light traps, staying and trap netting. And we collected the dominant consumers and the dominant resources in these wetlands, totaling over 500 samples that we're going to be running in stable isotope analysis. Stable isotope analysis, a common phrase you hear is, "you are what you eat," and I'm going to continue to use that example. So here I picked the flat iron steak for dinner tonight, so I am part cow. But you have to remember that cow is also a consumer, so we have to think about what is the cow eating. Let's say it's 100% grass-fed, so I would also be part grass. Let's say the cow was actually fed grass and a grain diet. Well, those resources are ultimately supporting the upper consumer and ultimately supporting me. Stable isotopes can also shed light on how much of the grass or grain are contributing. So if it was a 50-50 split or 80 grass, 20 grain, we can start to uncover that. So before I dive into data, I want to orient us all in isotope space. And we'll start with our carbon-13 on the x-axis, which is our sources in these wetland habitats. Our nitrogen-15 is our y-axis, which is going to be our trophic position. So as we increase in nitrogen, we increase in trophic position. You're going to hear me talking about enriched and depleted. Enriched sources of carbon are going to be on the right, depleted on the left. Enriched nitrogen are going to be higher on the plot, depleted lower on the plot. So if we add some sources in here, and let's say our primary consumers, we have zooplankton feeding on cestin and some isopods feeding on detritus. So the cestin is supporting the zooplankton, the detritus is supporting the isopods, they're falling in line. If we add some more consumers, in this example, we would say because they're falling in line here under cestin, then they are ultimately being supported by that cestin. So the zooplankton are feeding on it, forage fish are feeding on the zooplankton, and then pike feeding on that. But if we have an example where it's like this, where we have pike somewhere in the middle, well now it's relying on sources, on both of these sources, a mixture of these two sources. Remember, pike are generalist feeders, and so especially at this YOY stage, they could be eating both isopods and our forage fish, and then those resources, those basal resources below are supporting. And then there's not just two sources in these environments, there's multiple sources. A lot of times sources will be very close to one another where it's difficult to distinguish them. And in these cases, we're just going to group them together and make note of it and then continue on with the analysis. All right, so getting into the data that we have. This first graph, these first plots that I'm going to show you is all of the resources we have collected and analyzed so far for all 7 wetland sites, or habitat sites. It's about 200 of the 500 plus samples that we have. To start off, wetlands are at the top, bays are at the bottom. Don't worry too much about the colors and what the sources mean. For this plot, the biggest thing we want to talk about is we're seeing this separation of wetland sources being a little more depleted to the left compared to our bay sources, which are a little more enriched to the right. We're also seeing in our wetlands the sources are a little more grouped together, whereas in the bays they're further spread apart. So what this can mean, this possibly can mean is that we can start to discriminate spatially consumers based on their isotope values. If you have a very depleted pike, perhaps it's coming from a wetland. If you have a very enriched pike, perhaps it's coming from a bay. If we go and look closer at individual sites, one in the wetland, one in the bay, I've taken our sources and condensed them into their means and standard deviations here, and I've also included our northern pike for each wetland site, or each site, wetland or bay. And that shaded area is the convex hull, or equivalent to trophic niche area. So the thing that jumps out, and I think the thing that's most exciting about this, is we're seeing the trophic niche area of the wetlands is greater than in the bays. So hinting at some individual specialization of the feeding in the northern pike in these wetlands. Additionally, like the resources as a whole, our sources here are more depleted than the ones in the bay, being more enriched to the right. And we're also seeing in the bay, these sources are more spread out. So before we even run mixing models, we might start to infer what resources are supporting our northern pike here. The northern pike are further to the right, so they're being supported more by this pink one, which is macroalgae, purple is fine particulate organic matter, orange is detritus, and blue is macrophytes. So as to why we're seeing this increase in trophic niche area, there's a few things we can think about. Perhaps this is intraspecific competition. There are more pike in these wetlands than in the bays. Perhaps it's interspecific competition between chain pickerel and grass pickerel that live in these bays as well, or these wetlands as well. And so that results in, as they're competing, they have to use suboptimal resources, a more wide variety of resources to grow and develop. Now additionally, there is another topic that might explain this variation and this increase, and that's the microhabitat. If you recall earlier, we talked about the connectivity channels going through and the spawning pools that were created in efforts to restore these wetlands. But there's also areas that were untouched in the restoration projects. These are reference channels that the pike still access and spawn. So what we see here, we collected pike in both of these habitats, and we're starting to see this potential grouping of spawning pool pike, the ones that spawn or develop in here, through there, are more enriched in carbon and in nitrogen compared to the ones that are in the reference channels down through here, which are grouped in this blue, which are more depleted in carbon and nitrogen. So perhaps this microhabitat is resulting in different uses of resources to the pike and ultimately forming these groups. This is only 10 points and one wetland site. I have 3 more sites to look at. So it'll be interesting to see what comes out of that. The big variability that you see in these sources could also potentially be explained because we have these different microhabitats as well. So what's next? Well, the biggest thing is to finish processing and analyzing my samples, as well as including primary and secondary consumers into our isotope biplots, so that we can then run our mixing models to actually get the contributions of sources to our northern pike. And then we have environmental data, water quality data, as well as our vegetation and fish community data that could help us further distinguish these two wetland types, the wetlands and the bays, these habitat types. So what did we learn? Well, geomorphic types show separation in isotope values, which could lead to us being able to distinguish pike between these habitats using isotopes. The preliminary results suggest a difference in resource uses of northern pike between the bays and the wetlands, with the wetland sites having more individual specialization. And then as we're starting to do mixing models, we see that those wetland sites are really grouped together in their sources, and it might be difficult to separate them into groups for our mixing models. So we're going to have to run those and see what happens, essentially. And just as basal resources are the foundation to our food webs, this work and this study is very much foundational for further studies on upper trophic support in nursery habitats. Really great follow-ups to this would be looking at the nutritional value of these resources to pike and to the food web or the effects of invasion on the food web. All of my sites are impacted by this invasive cattail, and so looking at sites that don't have that cattail present could unveil some things. Also looking at the effects of environmental stressors on food webs. If that's of interest to you, stick for the next talk, because my lab mate Ben will be talking about hypoxia and its role in the northern pike's diets and in the food web. This also can help us inform management decisions that involve nursery habitats, especially around the resources that are supporting them. And we just further understand how the basal resources support a tertiary consumer in a nursery habitat. I'd like to thank New York Sea Grant and our partners in this project, as well as the Thousand Island Biological Station, and all the techs that work at the biostation for helping us with our broken boat motor when we flip or sink canoes in the wetlands. We wouldn't be able to get all that we got done without their help. And with that, I will take questions.
We have time for one, maybe two questions. Yes.
What would be an ideal plant community in a wetland or swamp, in absence of the
An ideal habitat?
plant community, like types of plants?
Yeah, so they prefer